In midlife and beyond, the enzymatic vacuum cleaner known as CD38 accelerates its consumption of NAD+, the redox currency that powers mitochondrial repair, sirtuin activity, and genomic maintenance. As NAD+ is drained, mitochondria become inefficient, reactive oxygen species rise, and the nervous system struggles to sustain stable sleep and circadian rhythms. By 2026, Apigenin, a flavone concentrated in chamomile and parsley, has become a widely used CD38 inhibitor and sleep molecule, but the rigorous human data behind those claims are far more fragile than popular narratives suggest. Disclaimer. This content is for educational purposes only and does not constitute medical advice. Always consult a healthcare professional before beginning any new supplementation or longevity protocol. The most detailed data regarding CD38 and Apigenin still come from mechanistic work published in the Journal of Biological Chemistry, which demonstrated that Apigenin directly inhibits the enzyme and raises intracellular NAD+ in mouse and cellular models. In obese mice receiving Apigenin, hepatic NAD+ levels increased, global protein acetylation decreased, and glucose and lipid homeostasis improved. These benefits disappeared in CD38 deficient models, confirming a specific dependent mechanism. A subsequent study on diabetic renal injury published in the journal Aging confirmed this, showing that inhibition by Apigenin restored the NAD+ to NADH ratio and activated mitochondrial SIRT3 in rodent models. Crucially, there is still no published human trial in which isolated Apigenin was administered, CD38 activity in the body was measured, and NAD+ concentrations were quantified with statistically significant p-values. A 2024 Systems Biology paper on NAD+ discusses Apigenin as a promising inhibitor within a broader network, but it does not yet present human interventional data for the molecule alone. Across 2024 to 2026, the literature contains no randomized human trial directly testing isolated Apigenin as a CD38 inhibitor. Popular science sources summarizing the story accurately reflect that inhibition of CD38 can elevate NAD+ in preclinical systems, but they also acknowledge that the specific magnitude of NAD+ changes in humans is currently unknown. The human sleep data also do not involve isolated Apigenin. Instead, researchers almost exclusively use chamomile extract. A key randomized controlled trial in chronic insomnia gave adults 540 milligrams per day of chamomile extract for 28 days. The results showed no statistically significant improvement in objective sleep measures, including sleep latency and total sleep time, compared with a placebo. While subjective sleep quality often improves in pilot studies, detailed statistics for deep sleep duration or REM proportion are not available at a level that would support a mechanistic claim for Apigenin. Given this gap between mechanistic enthusiasm and human trial data, a logical, modular framework for Apigenin can be described as a series of biological optimization layers. Layer 1 focuses on foundational NAD+ maintenance. This relies on well-supported behaviors such as consistent sleep, resistance exercise, and time-restricted feeding, which reduce NAD+ consumption at the source. Layer 2 views Apigenin as an experimental adjunct. While CD38 inhibition is well documented in animals, it remains unproven in humans in terms of clinical benefit. For those choosing to experiment, common practice involves 50 milligrams taken 30 to 60 minutes before bed, though some individuals escalate to 200 milligrams. Layer 3 addresses sleep and anxiety modulation. Preclinical experiments suggest that Apigenin binds to GABA-A receptors and exerts mild sedative effects, providing a plausible path to improve sleep initiation. As of 2026, critical questions remain unresolved. It is unknown whether common supplemental doses meaningfully inhibit CD38 in humans, or if such a change would translate into improved functional outcomes. Until those studies are performed, Apigenin should be regarded as a mechanistically compelling candidate for NAD+ modulation, but not yet as a molecule with confirmed human clinical results. Disclaimer. This content is for educational purposes only and does not constitute medical advice. Always consult a healthcare professional. For the complete clinical protocol summary, dosing spreadsheets, and our lab-vetted sources, visit the Longevity Logic Labs Protocol Hub linked in the description below. We update this database in real time as new clinical data and purity batches are verified. Thank you for supporting evidence-based science. Subscribe to stay updated on the latest longevity logic.